Now we turn to our last presentation. The organizers of the Congress for many years have had two senior awards, one that we had originally given to Eduardo Tolosa, named for Stanley Fahn, and the second is named for C. David Marsden, and this is a basic science award. And the winner of this award this year is close colleague Dr. D. James Surmeier from Chicago, from the United States. And the title that Jim has chosen is From Causes of Selective Neuronal Vulnerability in Parkinson's Disease to a Phase 3 Clinical Trial. So welcome, Jim, and please give him your attention.
Well, I want to start by thanking the committee of the meeting for this great honor. Really, for a basic scientist, this is really something because I think it's so important, the work that the members of this society do for Parkinson's disease patient, movement disorders patients worldwide. So I really want to thank the selection committee for this honor. I want to start with this slide. This is really the challenge that we all face. I mean, I'm clearly standing over on this side of this gulf, and I actually have a disclosure to make that's not in the book. Much to the chagrin of my mother, I'm not an MD, I'm just a PhD. In the program I'm listed as an MD PhD. I'm really a basic scientist. And we face this gulf between where we are, where our current understanding of disease mechanisms are, brain function, and clinical practice. And I can't understate the importance of the dialogue between basic scientists and clinicians in achieving any kind of translation. There are many, many people in this society who've really helped me bridge this gap. Tanya Samuni is a movement disorders colleague at Northwestern University who's been a partner with me all the way through the effort to bring our ideas to clinical trials. Also, many other members of this society, two of whom are here, Glenda Halliday and Jose Obiso, have also been instrumental in helping to formulate my ideas about the disease. It's also a particular honor for me to be associated with David Marsden, who was obviously a great neurologist. But one of the things Marsden was committed to was the idea that the best way to develop new therapies for diseases was to understand their causes, not just to use phenotypic models to screen drugs. And so, with that in mind, you can ask the question, what causes Parkinson's disease? Obviously, it's the subject of a lot of discussion at this meeting, not just this year, but for many of the years preceding it. What we know is that age is a factor. We hear repeatedly that genetic mutations are a factor. Environmental toxins are a factor. They've all been implicated in pathogenesis of Parkinson's disease. But those factors don't tell us something very important about the human condition, about the human pattern of pathology in Parkinson's disease patients. Now, we know that there are two key features of the pathology in Parkinson's disease. One is Lewy pathology, alpha-synuclein aggregations. The second is neurodegeneration. So this is work of Glenda Halliday's, mapping out the pattern of Lewy pathology in Parkinson's disease patients as a function of stage, in the pre-symptomatic phases and when symptoms begin to emerge, and then the later stages of the disease. And one of the things you can see is the Lewy pathology has a very distinctive pattern, even though not all of the patients conform to this pattern. This is the most common pattern, where it begins in the caudal medulla, and then it begins to appear more rostrally, and then finally in a more distributed fashion in the telencephalon. Okay. Now, one of the key things about this pathology is it does not spread from its site of initiation, regardless of where that is, meaning it doesn't spread to nearest neighbors. So this is, again, some maps that Glenda created where red shows you where the Lewy pathology is, and then in green are neighboring structures that never manifest Lewy pathology. So Lewy pathology does not spread from the DMV to neighboring structures. And this is also true in the mesencephalon, in the diencephalon. It's also true in the telencephalon. So this tells us that Lewy pathology does not simply spread in a non-synaptic or random way through the neuropil. So what causes that distinctive pattern of Lewy pathology? All right, now, you've heard many, many times at this meeting that Lewy pathology spreads. And the most common theory about spreading is that it's synaptic. The misfolded alpha-synuclein spreads from one neuron to another at synapses, and it spreads in a retrograde fashion, suggesting that the connectome of the brain is the determinant of that specific pattern of Lewy pathology we see in Parkinson's disease patients. So the question is that assertion, although it seems self-evident, is it consistent with the data? Now we have techniques for doing non-biased, systematic mapping of the connectome. This is one of the consequences of the initiative in the States to map the brain. All right, so one of the techniques is the monosynaptic rabies virus mapping technique. This will tell you all of the cells that are connected to a genetically identified population of cells in the brain. And so there are several papers that have been published recently. Not all of the structures involved in PD have been mapped, but a few of the key ones have been. So we can look at that literature and begin to test the idea whether or not Lewy pathology spreads transsynaptically in a retrograde fashion. So here's a map. I've taken two structures, the locus coeruleus and the substantia nigra pars compacta. And what we can do is we can look at the connectome. The size of the magnitude of the connection is coded by the diameter of the sphere here. So we would predict that structures that had strong synaptic connectivity with the locus coeruleus, which clearly manifests Lewy pathology, would have a high probability of manifesting Lewy pathology themselves. Structures that had an intermediate level of connectivity would have an intermediate probability of manifesting Lewy pathology, whereas those structures that were weakly or not connected with the locus coeruleus would have no probability of Lewy pathology. The other thing that's coded here is neurotransmitter phenotype. Okay. So this is the data from the locus coeruleus mapping. It clearly is not consistent with a synaptic spreading hypothesis that is based solely upon connectivity. That is, the cerebellum and the medial reticular nucleus of the medial reticular nucleus are the two most prominent synaptic inputs to locus coeruleus. Neither manifests Lewy pathology. PAG doesn't. Anterior nucleus of the amygdala doesn't. Whereas other structures do. The other thing that's interesting about this map is that GABAergic neurons, which are coded in purple, almost never manifest Lewy pathology, regardless of their synaptic connectivity. Well, maybe this is something peculiar about locus coeruleus. Let's look at substantia nigra pars compacta. This is the map for compacta. Again, it is not consistent with this prediction. We've known for a long time that the major synaptic input to substantia nigra pars compacta is from the striatum. And while there are Lewy neurites early in the disease that are a reflection of degenerating dopaminergic fibers in the striatum, there's no postsynaptic Lewy pathology there. Okay. Another major target of the substantia nigra is the globus pallidus. Again, no Lewy pathology. So if in fact there is spreading, and I cannot eliminate the possibility that there's spreading, there must be cell autonomous factors or regionally autonomous factors which govern the spread of that pathology. That is, if there's a seeding event here, there are some synaptic partners of this cell which will never manifest Lewy pathology and others which may. Okay. So the question is, what are those factors? Now, one of the other things I want to emphasize here is that we often equate Lewy pathology with neurodegeneration. Again, Glenda's data suggests that that's not the case. that in fact neurodegeneration evolves with a very different pattern than Lewy pathology. So again, this is the site of degeneration in the pre-symptomatic stages. There's loss of neurons, dopaminergic neurons in the ventral tier of the substantia nigra before symptoms emerge. In the symptomatic phases, the degeneration spreads out or is found in other regions, but it's not caudomedulla. The degeneration there only appears very late in the disease. So again, there's a dissociation between neurodegeneration and Lewy pathology in the human condition. So the take-home message here is that Lewy pathology does not inevitably lead to neurodegeneration, and neurodegeneration can occur in the absence of obvious Lewy pathology. And of course, that doesn't mean that there's not synuclein pathology, but there is not Lewy pathology, which we have an unequivocal measure for. All right. So if it's not Lewy pathology, what is it? Now, one of the things we know about the brain is that it has enormous heterogeneity in the cells that are in the brain, both anatomical, which is shown here, as well as physiological and biochemical. Our prediction is that neuronal phenotype is actually a predictor of pathology in the disease, so that some neurons respond to the challenges posed by Parkinson's disease, again, whether it's age, genetic mutations, or toxins, by continuing to function normally. Whereas other cells, that same challenge leads to pathology and death. Okay, so the important thing about this from a therapeutic standpoint is that if we know what that neuronal phenotype is, it's possible that we might be able to alter that phenotype, and in so doing, alter the pathogenic course of the disease without doing anything about age, genetic mutations, or past exposure to environmental toxins. So what are those characteristics that might allow us to alter the course of the disease? Now, our work started with the study of dopaminergic neurons in the substantia nigra. We all know that those cells are the ones that govern the appearance of the motor symptoms of the disease. Again, this is a slide that you've all seen many, many times, where the normal pigmented dopaminergic neurons in the mesencephalon of a healthy patient are lost in a PD patient. So what are the phenotypic features of those neurons that might cause them to be vulnerable to neurodegeneration and to Lewy pathology? Now, one of the things about these cells that's been talked about a lot is the fact that they have massive axonal arbors, and that's absolutely true. This is some work of Matsuda et al. Showing a massive axonal arbor of a substantia nigra pars compacta neuron in the striatum. Its cell body is sitting way down here, and here's the axon that stretches up into the striatum. Now, it's very clear that that could create a proteostatic stress, or that might create a bioenergetic stress that could promote pathology. But there's nothing much we can do about that. Moreover, we also know that there are other cells which have every bit as large an axonal arbor, but do not succumb in the disease. One of those is the cholinergic neuron in the striatum, which may have even larger axonal fields than those of the compacta neurons, and yet they never show signs of Lewy pathology or degeneration MPD. What are some other risk factors? One of the things that is a characteristic of dopaminergic neurons in the substantia nigra is that they're autonomous pacemakers, very much like cardiac cells. So they're spiking 2 to 10 hertz all the time, right? They do that in the absence of any synaptic input. They also have very broad action potentials, which contrast them with neighboring neurons which are resistant to the pathogenic mechanisms in Parkinson's disease. Now, that pacemaking is absolutely necessary to maintain dopamine concentrations in target structures of the basal ganglia like the striatum, which enable movement and action selection to happen. Right? So even transient suppression of that pacemaking activity can have effects on movement and the ability to respond to environmental stimuli. Now, as a consequence, that pacemaking mechanism is very robust. There are 2 pacemaking oscillators, one in the soma and axon initial segment, which use an oscillating series of ionic conductances to drive that pacemaking activity. This one is actually common to a lot of pacemakers in the brain. What's distinctive about compact neurons is that in addition to this very common pacemaking mechanism that relies upon monovalent cations, there's a dendritic pacemaker that involves calcium. In particular, L-type calcium channels play a very important role in this dendritic oscillator, which helps make the pacemaking robust. By that I mean resistant to perturbation. Now, this tonic activity makes these cells like leaky boats. Right? They're constantly spiking, constantly dissipating the ionic gradients which underlie electrical activity. This dissipation, this opening of ion channels leads to a lowering of the electrochemical gradients, that underlie excitability, which has to be restored with ATP-dependent mechanisms, pumps and exchangers. And as a consequence, this creates a bioenergetic challenge to these cells. In particular, these cells seem to be very close to a bioenergetic cliff. Most neurons don't have much respiratory reserve capacity. These cells seem much closer to that bioenergetic cliff than most neurons. Now, that, again, poses a major problem. Again, going off that bioenergetic cliff would silence these compact neurons, leading to a drop in dopamine concentrations within the striatum and an inability to move. This would be obviously a survival disadvantage in the wild, right? And so I think that there's been strong evolutionary pressure to maintain as robust a pacemaking mechanism in compact as possible. So calcium, again, plays a very important role in increasing the robustness of these cells and their bioenergetic challenge. Calcium, in particular, helps compact neurons move back away from this bioenergetic cliff. How do they do that? Well, I'm not going to go into all the details. This is one of the things that we spend a lot of time studying. But calcium coming in during the pacemaking process induces release of calcium from intracellular stores, which leads to the injection of calcium into mitochondria, which stimulates the TCA cycle and drives oxidative phosphorylation, increasing the production of ATP. This is very, very much like the mechanism that we know exists in the cardiac cells and in skeletal muscle, which help us maintain the ability not only for our heart to continue beating, but also for muscles to continue to contract in response to prolonged exercise. It helps the cell anticipate the bioenergetic needs without ATP levels falling. So this is a very common mechanism. So the question is this really important in neurons of the substantia nigra? So we did a little test. All right? We can imagine being chased by this tiger. How do we can mimic that with the cells by elevating extracellular potassium to increase the discharge rate. Now, normally, compact neurons respond to that challenge by continuing to spike for as long as we can watch them. But if we take away L-type calcium channels and redo that experiment, what happens is after a very short period of time, the compact neurons fail. And the tiger or the lion in this particular case catches us as we fail to be able to move quickly. So again, I think that there's been very strong evolutionary pressure to maintain this mechanism where calcium channels, in particular L-type calcium channels, play two very important roles in the life of a compact neuron. One is they are electrically important. They help maintain the oscillatory behavior of those cells, which continues to release dopamine in the striatum and the rest of the basal ganglia. The other thing calcium does is it stimulates mitochondrial oxidative phosphorylation and boosts ATP production to make sure that that activity continues. This, of course, increases the robustness of pacemaking, again, by maintaining ATP levels in the cell. All right. The problem is that design comes at a cost. What else in life that's good doesn't come at a cost? So in this particular case, oxidative phosphorylation is capable of generating potentially damaging superoxide and reactive nitrogen species. And in fact, if we use a mitochondrially directed probe to monitor oxidative phosphorylation and redox stress in compactor neurons, what we see is that in relatively resistant ventral tegmental area dopaminergic neurons, there's no measurable oxidant stress. But there clearly is in dopaminergic neurons that are pacemaking, that are doing their job, okay? So this is simply a consequence of that feed-forward mechanism that's driving oxidative phosphorylation all the time with every cycle of the pacemaking. What's important from a translational standpoint is that oxidant stress can be alleviated. And it's alleviated by inhibiting the L-type calcium channels that I've just talked about. Isradipine, which is a dihydropyridine inhibitor of L-type calcium channels, almost completely eliminates the oxidant stress seen in mitochondria of compactor neurons. Now, one of the things I want to emphasize is although I've spent a lot of time talking about substantia nigra, this same phenotype is seen in almost all of the cells that have been looked at carefully that manifest either neurodegenerative potential or Lewy pathology in the disease. It's true of DMV, it's true of locus coeruleus, it's true of dorsal raphe, it's true of the pedunculopontine nucleus, it's true of basal forebrain. Those cells all have a common phenotype. They're all neuromodulatory systems that tend to mobilize motor and sensory pathways that are involved in action, selection, and escape from prey or in predation. You have a very distinctive phenotype, which again includes not just its pacemaking, but a large axonal field. Okay, so again, what I've initially tried to say was that neuronal phenotype might interact with all the things that we know are involved in PD pathogenesis to drive this specific pattern of disease. So let's dig down just a little bit deeper in this and say whether or not the characteristics that I've just told you agree with what we know about pathogenesis. What we know is that these factors drive two major pathogenic mechanisms. One is mitochondrial dysfunction, which you just heard about. The other is proteostatic dysfunction, that is Lewy pathology. Those two things lead to pathogenesis. What I've told you is that the at-risk phenotype has elevated mitochondrial stress. It also has elevated cytosolic calcium, as well as a large axonal arbor. All three of those factors drive both of these pathogenic mechanisms. Sustained mitochondrial oxidant stress leads to mitochondrial DNA deletions, which impair mitochondrial function. Mitochondrial stress also can impair proteostatic function. Elevated calcium can clearly impair proteostatic function and also promote alpha-synuclein aggregation through activation of calpain, C-terminal cleavage, as well as misfolding of alpha-synuclein. If you want to misfold alpha-synuclein and form fibrils in a dish, what do? You add calcium. The other thing is that the large axonal arbor could certainly feed into both of these pathogenic mechanisms, again, helping to accelerate or intensify the pathogenic mechanisms in this specific subset of cells. Okay. From a translational standpoint, the key question is, can we re-engineer those cells to make them less vulnerable, to slow the progression of the disease? Dihydropyridines have been in clinical use for a long period of time for the treatment of hypertension. They have very good brain bioavailability. They get into the brain and could target these channels. One of the things that we know is that inhibition of those L-type calcium channels, although it decreases the robustness of pacemaking, it does not prevent pacemaking from happening. So this is just a recording of a compactor neuron. We're showing the dendritic calcium oscillation. If we apply a dihydropyridine, we can eliminate the dendritic calcium oscillation. We can eliminate that feed-forward bioenergetic control mechanism without changing the rate and regularity of pacemaking. So from a clinical standpoint, it's a viable path forward. If it were to stop pacemaking, obviously it wouldn't be a viable path. In fact, dihydropyridines make vulnerable compactor neurons very much like relatively resistant ventral tegmental area neurons, which pacemake but have no dendritic calcium oscillations. The question is whether chronic inhibition of CAV1.1 channels, 1.2, 1.3 channels is a viable therapeutic strategy. Now, clearly, we don't face this kind of pressure anymore, right? If we did, it wouldn't be a viable path forward, but we don't. The question is whether or not there are adaptations to chronic administration of dihydropyridines which would defeat the purpose of drug administration. Now, we know that in many cases, this is what happens. You inhibit a protein and the neurons, or whatever cell type you're thinking about, compensate by upregulating that protein or a companion protein that has the same function. So when we've done this, we've looked in animals, adult mice, where we've chronically administered dihydropyridines with subcutaneous pumps. This just shows a normal pacemaking with a normal dendritic calcium oscillation. If we acutely inhibit L-type calcium channels, it doesn't affect pacemaking, but diminishes the calcium oscillation and mitochondrial oxidant stress. If we look at what happens in the chronically administered case, it's beautiful from the translational standpoint. There's a very clear diminution in the dendritic calcium signal, again, without any change in pacemaking. And this is very important to us because germline deletion of CAV 1.2 or 1.3 channels leads to an upregulation in another class of calcium channels, and there's no change in the dendritic calcium oscillation. What about mitochondrial function? Chronic treatment lowers mitochondrial stress more than acute antagonism of CAV1 channels. And you can see that here. What we're doing is looking at mitochondrial oxidant stress in compacted neurons. Normally it's high. Acute treatment with isradipine knocks it down. Chronic administration of isradipine lowers it even more, which was a bit confusing to us at the beginning. Why should that be? Until we looked at mitochondrial mass in these cells. So normally mitochondrial mass in compacted neurons is about 20–30% lower than it is in most of the other neurons in the brain. This is just the green here are the mitochondrial labeling. This is with a mitochondrially directed GFP variant. This just shows the cytoplasm, which is stained with a tyrosine hydroxylase stain, and the nucleus, to give you an idea of where these different compartments are. In a chronically treated compacted neuron, what you can see just by eye is there's more green in the cytoplasm than there is in the naive cells. And on average, mitochondrial mass rises by about 30% with only 10 days of isradipine treatment. Now, why that is, we're not entirely clear. We think that the mitophagy mechanisms that you just heard about are slowed because the damage we're doing to mitochondria is less. That is, we've diminished their oxidant stress, which is one of the major drivers of mitophagy and mitochondrial damage. But that remains to be determined. Okay, let me finish by trying to bring this back to the human condition. And what I've shown you so far is basically all from mice, not from man. This is where we want to be. So does what I've told you have anything to do with the human condition? Now, we don't have an animal model with good predictive validity for neuroprotection. This is one of the shortcomings in the field. So there's no way for us to really test our hypotheses in mice and beyond what I've shown you. All right? But there is data out there. And again, this is the bridge between the clinician and the basic scientist. When we first made the suggestion that dihydropyridines might be neuroprotective, people said, well, I mean, come on, they've been in therapeutic use, clinical use for a very long period of time. We'd have known, but nobody went out to look. And so there are 4 major epidemiological studies that have been done so far. All of them say the same thing. And that is, if you were treated for hypertension with dihydropyridines, as opposed to ACE inhibitors or beta blockers, your risk of developing Parkinson's disease drops significantly, by 30% or more. So that was a great indication. The other thing that we know that I'm not going to show you is that the cells that are lost in humans are cells that lack calcium buffering capacity. They don't express calbindin, which is, again, consistent with the general hypothesis. The other thing is we see massive accumulations of mitochondrial DNA mutations, or deletions, excuse me, in the mitochondria of compacted neurons in Parkinson's disease patients, as well as with normal aging, but more importantly, more dramatically in Parkinson's disease patients, which is again consistent with the idea that there's sustained mitochondrial oxidant stress in compacta. But really, there's only one way of testing this hypothesis, and that is to do the human experiment. Dihydropyridines are off patent. There was no drug company that was interested in doing this, so it fell upon the back of the government in the United States. In 2013, NIH funded a 5-year, $23 million, 56-center phase 3 clinical trial. Tanya Samuni is the leader of this trial with help from the Parkinson Study Group. Again, this is again a partnership between basic scientists and clinicians. This just shows all the performance sites. The study will be completed in 2018. There are so many centers involved in this because the patients enrolled had to be just recently diagnosed. This is an intervention that we think will have its greatest impact early in the disease. So let me summarize. What I've tried to convince you of is that vulnerable neurons share a common phenotype, and one of the key features of that phenotype is autonomous pacemaking. They're constantly active. That the engagement of L-type calcium channels, CAV1 channels, makes that pacemaking more robust. It does it through both electrogenesis as well as bioenergetic feed-forward control mechanisms. Now, while that increases the robustness of pacemaking, which must have been a great evolutionary advantage to us and to other mammals, it comes at the expense of elevated mitochondrial oxidant stress. For short-lived animals, like rodents, there's no consequence to this because the impact of that elevated mitochondrial oxidant stress takes decades to manifest itself. Sustained inhibition of those channels increases mitochondrial mass and lowers mitochondrial oxidant stress without doing anything to the pacemaking, without having any obvious behavioral consequence. In humans, inhibiting those channels is associated with decreased risk as well as slowed progression of Parkinson's disease. And again, there is evidence, epidemiological evidence for that I didn't show you. That combination of basic science and clinical observation was what drove the clinical trial forward. It was that partnership that was essential to the success of our effort to convince the federal government to spend the money needed to run this trial. And that trial is ongoing, and we'll know the outcome soon. All right, so let me tell you about the people who really did this work. This was a team effort. A very dedicated collection of postdoctoral students and predoctoral students listed here were essential to this effort. Paul Schumacher, who's a faculty member at Northwestern, developed some of the transgenic models that we used, in particular the ones with mitochondrial redox probes. And again, Tanya Samuni is the director of the Movement Disorders Clinic at Northwestern, and she was really the one who drove the clinical effort here. Jose Obiso has been a long time discussing sort of collaborator. Glenda Halliday also has been very important to my thinking. They were responsible for some of the graphs I showed you in the very early stages of the talk. Serena Rizzuto and Tulio Pazan gave us probes. And of course, none of this would have been possible without financial support from both private and public organizations. So at that point, I'll stop and thank you for your attention.